For today, I'll be discussing to you chapter 11, which is all about the blood. For the characteristics of blood, first, it is a type of connective tissue found in your body. It is sticky, it's heavier than water. The oxygen content of your blood determines its color. The temperature is slightly higher than the rest of the body. For males, you have 5 to 6 liters of blood, and for females, it's around 4 to 5 liters. So by computing the percentage of blood based on your body weight and all other fluids and tissues found in your body, so for the blood, it's around 8% of your body weight, and then for other fluids, that's 92%. So for you to be able to segregate the components of your blood, you can actually draw blood from your body and check upon the percentage by volume of its components. So you usually draw blood and then use a centrifuge tube and then use a centrifuge equipment to um, separate its components. So 55% of your blood is actually made up of plasma. So this plasma, it includes your proteins, which are 7% in total. You also have 91% water and other solutes for 2%. So for the proteins found in your plasma, we have there 58% albumins, 38% globulins, and then 4% of fibrinogen. For the other solutes found, you have ions, nutrients, waste products, gases, and regulatory substances. And then for your formed elements, you have there your 45%, which is comprised of your platelets, around 250 to 400,000 of them. And then in between, you have the white blood cells, which is around 5 to 9,000. And they are made up of your white blood cells, including the neutrophils, which are 60 to 70% in number. You have your lymphocytes, 20% to 25%. Your monocytes, 3% to 8%. Eosinophils, 2 to 4%. And the basophils, 0.5 to 1%. And then the remaining components of the blood are your red blood cells, which is around 4.2 to 6.2 million in number. So for the composition of blood, first we have what we call again the plasma. It is 55% of the total blood. Its color is pale. It's yellow liquid that surrounds your cells. 91% is water, 7% is proteins, and 2% are other inclusions of it. And then you have the formed elements, 45% of your total blood. It includes your cells and cell fragments, including your erythrocytes, leukocytes, and your thrombocytes. For the plasma proteins, first you have your albumin. It makes up 58% of your plasma proteins. And it is actually a protein made by your liver. So it helps maintain water balance in your bloodstream so it doesn't leak into your tissues. Other plasma proteins include also your globulins, which happen to be 38% of your plasma proteins, which helps in your immune system. So the different types of your globulins include alpha globulins. So there are two types. You have alpha 1 and alpha 2. They have a little difference in structure, but they perform the same function in carrying hormones, cholesterol, and copper through the bloodstream and act as an enzyme for certain chemical reactions in the body. For alpha globulins, it also works to help or prevent the actions of other enzymes such as those that cause the blood to clump. And then we also have the second type, which is the beta globulins. This is similar in structure with the alpha globulins, but the only difference is that they do have the beta 1 and beta 2. So they carry lipids, hormones, and cholesterol through the bloodstream and assist immune cells in mounting an immune response to invading bacteria, viruses, and parasites. Next, we have your gamma globulins. So they are blood proteins produced by lymphocytes and plasma cells of the immune system when an immune response is needed. So almost all GABA globulins are known as immunoglobulins or also called as your antibodies. So these are globulins that help with immune responses and your immunity. So for the types of your gamma globulins, we have IgA, or the immunoglobulin A. This is the second most common human immunoglobulin in your serum. So it is secreted in milk and is also the most prevalent immunoglobulin in secretions like your tears, saliva, and your mucus. The second type would be your immunoglobulin D, or IgD. It is expressed on the surface of mature B cells, or the B lymphocytes, for growth. And the B lymphocyte functions in the humoral immunity component of the adaptive immune system of or by secreting your antibodies. So the next one would be IgE, or the immunoglobulin E. It is primarily used for defending against parasitic invasion and is responsible for any allergic reactions. And then for IgG, the immunoglobulin G, the only class of immunoglobulin that can cross the placenta in humans, and it is largely responsible for the protection of the newborn during the first months of life. And then you also have the IgM, or the immunoglobulin M. It acts to quickly recognize and initiate an immune response by directly neutralizing pathogens or clearing novel antigens. So I know in this current situation that you have already read and heard more of these immunoglobulins that are being tested um, for ECLIA, to be able to check on the number of antibodies we have in our body, and to be able to know if you're somehow affected or not by coronavirus. But then again, there are researches and even big hospitals or medical associations that do not really support the use of the immunoglobulin testings, kasi they are not as accurate as the PCR. Okay? So for the plasma proteins, you also have your fibrinogen, which is actually 4% of the plasma proteins, and it aids in your clot formation. So in the process of the formation of clots, okay, you have actually different clotting factors. So these clotting factors, of course, do have several roles that they perform, and there are actually around 18 types of clotting factors. So please be please memorize, or at least know some of these clotting factors, because they may be asked during your exam. So what type of clotting factor is clotting factor 1? For example, fibrinogen. You have clotting factor number 2, or prothrombin. Okay, so apparently all of these clotting factors do have their specific roles when it comes to the clot formation. So this is actually the fibrin synthesis cascade. So again, fibrin is actually used, or it is a protein that's used when it comes to the formation of your blood clots. So there are actually two ways on how your blood clots can actually be formed. So first, you have your intrinsic pathway, wherein you have your damaged vessel wall. So there you will see that the clotting factors, okay, like 12, it will be synthesized to 12A, and then 11 will be synthesized by 12A to be 11A. Okay, so those are the complexes. And then you also have the other one, the extrinsic pathway, or the trauma to extravascular cells. So there you have your clotting factor 3, and then clotting factor 7 will be synthesized by the clotting factor 3 to become 7A. Okay, and then as you see on the image here, there's actually a joining complex from the intrinsic and the extrinsic pathway. So there you will see that the complexes will be actually combined in a certain level. So for them to be able to produce the clotting factor 10, and then it will be synthesized again by the clotting factor 5. So in the common pathway, So you have prothrombin 2 that will be synthesized to thrombin 2A, and then fibrinogen to fibrin, and then of course your and then it will be synthesized by 13A. So that's already the production of your cross-link fibrin clot. Okay. So the circles here that you see are actually factors in inactive state, and then they will be inactive state after they are being acted upon by the different clotting factors. Okay? So next, let's go now to the erythrocytes, or what we usually call as the red blood cells. Again, from the previous lectures that were already introduced to you, the common characteristics of your erythrocytes are this shape with thick edges, and it doesn't have a nucleus. It is actually lost during the development. It lives around uh, in a span of 120 days, and of course, the major function of your erythrocytes or RBCs are to transport oxygen to your tissues. Okay? So hemoglobin. So this is the main component of your erythrocytes, which actually transports your oxygen. So they are the ones responsible for the intake of oxygen of your red blood cells. So each globin protein is attached to a heme molecule, and each heme molecule contains one iron atom. So oxygen binds to the iron, and then you produce now what you call your oxyhemoglobin. This is the hemoglobin with an attached oxygen to it. Okay. So si heme, it's actually a molecule that's responsible naman for the iron, 'di ba? Kaya nga kapag low blood kayo, more likely ang pinapainom sa inyo ay iron supplements, okay? Because this iron promotes the heme attachments to be able to bind to oxygen, okay? So for the production of the erythrocytes, so decreased blood oxygen levels cause your kidneys to increase the production of what you call your erythropoietin, which is a type of hormone. So the erythropoietin stimulates your red bone marrow to produce more erythrocytes, and the increased erythrocytes cause an increase in blood oxygen levels. So the more RBCs you have, of course, the more oxygen you have in your body. So this is actually just an example of how your red blood cells are being developed or being reproduced. So decreased blood oxygen would stimulate your kidneys. So in response to the decrease of the oxygen, the kidneys would increase its erythropoietin secretion. So this erythropoietin would directly affect your bone marrow, and then your bone marrow will eventually increase the production of your red blood cells, and of course, the more red blood cells that you have, you will be able to increase your blood oxygen in your body. Okay? So the fate of old erythrocytes and hemoglobin. So what if dumating na sa time na kumbaga nag-old age na rin yung ating red blood cells. 'Di ba nga ang mga cells natin, they are being regenerated to new, and then to kumbaga sa development din ng tao, merong newborn, merong mid-age, merong old age, okay? So, old RBCs are removed from the blood by macrophages in your spleen and your liver. So, the hemoglobin is broken down, and globin is broken down into amino acids. And then, the hemoglobin's iron is recycled. The heme is converted to bilirubin, and bilirubin is taken up by the liver and released into the small intestine as part of your bile. So that's how your old erythrocytes and hemoglobin end up after um, being eaten by macrophages and being broken down into different components in your body. So this is the hemoglobin breakdown. So again, the first one, it's actually the work of the macrophage, okay, to eat up the hemoglobin, and it's broken down into individual amino acids. It can also um, produce or release the heme for iron, and then going to the bone marrow. And then the heme can also become bilirubin, and then bilirubin to your liver, to your intestine, and of course it also um, it's also being used in your kidneys. Okay, so just study how this hemoglobin breakdown happens. So next we have your leukocytes. So leukocytes are what we call also your WBCs, or white blood cells. So in this one, their difference with the red blood cells is that they lack hemoglobin. So they don't really have hemoglobin on them. So they are larger than the erythrocytes, and they contain nucleus. So the functions of the leukocytes are more likely to fight infections and to remove dead cells and debris by phagocytosis. So as you see here, we have actually two types of leukocytes. We have the agranular, so based on their features, they don't really have granules within their cytoplasm. So all you see there is that they have what you call the nucleus, and then you have the cytoplasm, and of course the cell membrane. So as you see here also with the granular leukocytes, the shape of their nucleus are different from each other. So here in the agranular leukocytes, you have your lymphocytes, and then you also have your monocytes. And then for the granular ones, you have here your eosinophil, the basophil, and then the neutrophil. So all of them are actually granular leukocytes. So you see that they have dots. So those are the granules found in their cytoplasm. So the types of the leukocytes, again, we have the granulocytes, which contain the granules. So the first type would be the neutrophil, which is the most common one. It remains in the blood for around 10 to 12 hours, then moves to your tissues. And they actually have the ability to phagocytize, or um, to eat up other components found in your body. And then next, we have your eosinophils. They are usually present when there's inflammation, so their function is to reduce them. And then you have the basophils, which are the least common, and they release histamine and heparin. So as you know, histamines do actually um, happen or released kapag ka meron kayong mga allergic reaction. Di ba nga, ang mga antihistamine, sila yung mga nagsusuppress ng inyong allergic reaction. So for the agranulocytes, these are the leukocytes that do not have any granules on them. The first type would be your monocyte. This is the largest type of leukocyte, and they produce macrophages. And then the second one would be your lymphocyte. It has immune response role. And then there are several types like your T cells and B cells, and they lead to the production of your antibodies. So in our, or if you check on the different types of leukocytes under the microscope, they look like this. Okay, so you see there that they do have really different types of, or different shapes of their nucleus, and which ones are granulated and agranulated. And then you also see there that there's also a difference when it comes to their color. Okay. So next we have your platelets. So in this image, you will see here that you have the red blood cells. This is actually how they look like under the microscope. So they are pink in color, circular, and then medyo faded yung gitna. Okay, and then you see here some examples of your white blood cells, including your lymphocytes and of course your neutrophils. And then yung mga nakikita nyo na mga blue na maliliit, those are your platelets. Okay, which are around the red blood cells and white blood cells. So they are actually blood clotting cells, and they are produced in your red bone marrow. So Hematopoiesis. So this is the process of the blood cell formation. In an infant, it occurs in liver, thymus gland, spleen, lymph nodes, and red bone marrow. And in adults, it occurs mainly in the red bone marrow. So basically, with infants, no, mas maraming nagaganap ng mga hematopoiesis. It's because it is aiding in the development and growth of infants. But when, of course, growth and development has already been reached, most of the hematopoietic processes are already done by your red bone marrow. Okay? And then, stem cell is actually what you call your original cell line. So, stem cells are actually, kumbaga, general cells sila, but then again, once na ang body natin ay nag-direct ng kanilang or nag-direct ng kanilang pinaka-role sa katawan, they can be changed into different types of cell. Pwedeng si stem cell mo ay maging cell, maging skin cell, pwede siyang maging muscle cell, pwede siyang maging red blood cell, pwede siyang maging white blood cell. So, kumbaga, ang ating mga stem cells ay nagde-develop depending on the need of the body and kung paano i-dina-direct ng katawan natin, kung alin yung may kailangan ng mga cells na yon. Kaya nga yung mga stem cell na yan, 'di ba, it is being talked about in the science industry because stem cells are being used na kumbaga to develop cures for cancer, 'di ba? Gusto nilang malaman if normal cells would be able to stop the growth of abnormal cells in cancers. Meron din naman na ginagamit yung mga stem cells to produce yung mga like sa mga animal models, 'di ba? Meron silang mga ginagawa na naglalagay sila ng tenga sa daga, kung posible ba yun. So, yun yung mga gamit usually ng stem cell. And for rich people, no, ginagamit nila tong stem cell na to make them look younger. Kasi again, guys, stem cells are actually the best types of cell na, kumbaga yung age ng cells na yun ay mga bagong produce ng katawan. So, young cells sila. Okay? So this one is actually how the stem cell is being, kumbaga, how a stem cell leads to different types of your blood cells. Okay? So from a stem cell, it can be a pro-erythroblast. And then it will develop eventually to an early erythroblast, and then develop into an intermediate erythroblast. And then you will have the late erythroblast. So in this stage, you see there that the nucleus has already been excluded. And then eventually, kapag nawala na yung nucleus na yon from the developing erythroblast, it will become a reticulocyte and eventually become a red blood cell. Okay? So for the granulocytes or the white blood cells, it will start from your stem cell and then it will become a myeloblast and then it will develop into a progranulocyte. So from a progranulocyte, it can be a basophilic myelocyte, it can be an eosinophilic myelocyte, or a neutrophilic myelocyte. So, kung mapapansin nyo dito guys, kapag basophilic, more likely color blue yung kulay niya. Kapag ka eosinophilic, ito yung mga medyo mapupula yung color nila. And then, kapag neutrophilic, usually, nagbabalansa yung color. Okay? And then, the basophilic myelocyte will then be developed into a basophilic band cell, and then eventually it will become a basophil. Okay? And then for eosinophilic myelocyte, it will become a eosinophilic band cell until it reaches to become a eosinophil. And then for neutrophilic myelocyte, it will become a neutrophilic band cell and then a neutrophil. So, dyan, yan yung pathway on how your granulocytes are actually developed. Okay? So next, for your lymphocytes or monocytes and or granulocytes. So From stem cell, it will become a lymphoblast, and then eventually it will grow into a lymphocyte. For the monocyte, it will become monoblast and then monocyte. So those are your agranulocytes. So again, remember that when you say blast, kapag may blast sa mga pangalan nila, yun yung mga types of new cells, right? And then for the platelets, so from stem cell, you have now your megakaryoblast, and then it will eventually become a megakaryocyte. And then the megakaryocyte breaks up to form your platelets. And so guys, this is actually an image to remember. Please know which are the precursors of each type of blood cell. So when you say precursor, ano yung naunang type of cell niya before it goes to the developed type of cell. So for example, ang tanong, what is the precursor of a progranulocyte? So the answer would be a myeloblast. Okay? So for example, what would be the precursor of a monocyte? So of course, the younger version or the precursor cell would be a monoblast. Okay? So that's um, how your different types of blood cells are being produced. And please make sure that you will be able to know the differences among the features of your basophilic, eosinophilic, and neutrophilic cells. Kasi kung makikita nyo dito, magkakaiba sila ng shape of the nucleus. Okay? So, blood loss. So, when blood vessels are damaged, blood can leak into the other tissues and disrupt normal function. Blood that is lost must be replaced by the production of new blood or by a transfusion. So, usually naman guys, kapag ang inyong mga sugat or injury ay maliliit lang, of course, the body can actually accommodate the replacement of the blood loss. But if it's something that's life-threatening, like kunyari sa pregnancy or yung talagang naputulan ka ng part ng katawan or nasaksak ka or may tumaga sa'yo, of course, it would need to have support by blood transfusion. So, diyan ginagamit usually yung mga blood na dinodonate natin sa blood bank just in case that a person would need to undergo transfusion. Okay, yan yung mga ginagamit nila. So, preventing blood loss. So, first you have your vascular spasm. This is a temporary constriction of your blood vessels. And then, the presence of the platelet plugs, which can seal up small breaks in your blood vessels. And then, the third one would be the blood clotting or the blood coagulation. So, for blood clotting, blood can be transformed from liquid to a gel. So, a clot is a network of thread-like proteins called fibrin that trap blood cells and fluid. Also, the clot depends on the clotting factors that you have in your body. And then clotting factors are proteins in the plasma. It is only activated following an injury. It is made from the liver and it requires you vitamin K. So this is actually how the blood clot or the general steps of clotting looks like. So first, you should have an injury. So the injury is when a blood vessel is severed and blood components are leaking out of the breaks. So as you see here in this image presented to you, of course, in your blood vessels, of course, the different types of blood cells travel along them, no? And then what would happen is that in the injury there will be a vascular spasm. So kumbaga magsisikip yung daluyan ng inyong blood vessel. So the vessel contracts near the injury point, reducing your blood loss. And then the third one would be the platelet plug formation, this part. Okay. So, platelets are activated by the chemicals released from the injury site and by contact with underlying collagen. The platelets become spiked and stick to each other and then in the wound site. Okay? So, ayan. Nagdidikit-dikit yung mga platelets. So, initial platelets are activated by chemicals released from the injured cells and by the in contact with the broken collagen. And bound platelets release chemicals that activate and attract other platelets. So, yung mga unang mga nagre-respond dun sa clot, nagtatawag pa sila ng mga kapwa nila na mga platelets to secure or to stop the or to um, kumbaga ignite or start the forming of the platelet plug. Okay? And then platelets move towards the source of the chemical signals and bind, and then platelet plug grows in size. So next, after the platelet plug, you already have your coagulation. So in coagulation, we have fibrinogen, which is converted to fibrin, which forms a mesh that traps more platelets and erythrocytes, producing a clot. Okay? So steps in clot formation, again, injury to a blood vessel causes inactive clotting factors to become activated due to exposed connective tissue or release of the thromboplastin. So prothrombinase or the clotting factor is a formed and acts upon prothrombin. And then prothrombin is switched to its active form thrombin. And then thrombin activates fibrinogen into its active form fibrin. And then the fibrin forms a network that traps your blood, or it's called the clot. So this is the clot formation. Okay, so again, you have the inactive clotting factors, and then there should be other inclusions as well, or presence of injury to the vessel, of course, the connective tissue exposed to chemicals released, and then it will become the active clotting factors. You have the calcium and platelet chemicals to produce prothrombinase, and then this one, prothrombin to thrombin, and then fibrinogen to fibrin. Okay? So, control of the clot formation. So, clots need to be controlled so they don't spread throughout the body. So, we have again anticoagulants that prevents clots from forming, like your heparin and your antithrombin. Okay? So, ito namang mga anticoagulants na to guys, usually, ito naman yung mga ginagamit kapag may sakit sa puso, di ba? Kapag merong mga clot formation na nagbabara sa mga ugat ng mga blood vessels, so yan mga anticoagulants ang mga ginagamit nila. And then, injury causes enough clotting factors to be activated that anticoagulants can't work in that particular area of the body. So, clot retraction and fibrinolysis. So, clot retraction is the condensing of the clot, and then serum in plasma is squeezed out of the clot and helps enhance healing. And then fibrinolysis, so again, when you say lysis, it's something that's broken down. So, it's the process of dissolving your clot. You have your plasminogen, which is a plasma protein that breaks down your clot, which is the fibrin. For blood reactions, injury or surgery can lead to a blood transfusion. So transfusion reactions or agglutination is the clumping of the blood cells. So you also have antigens. These are molecules on the surface of your erythrocytes. You have antibodies, which are the proteins in your plasma, and of course blood groups, which are named according to the antigen ABO. Okay, so ito yung determinants naman ng ating mga blood type. Okay, so for human, we usually have the ABO blood groups, but there are other animals that that do have their own blood type. Okay, hindi ABO yung mga ginagamit nila. But for us, it's ABO. So for the ABO blood groups, so again, for the type, you have the antigens. Ito yung A, B, A and B or none. Okay? For antibodies, you have antibody B, antibody A. It can be none, and then anti-A and anti-B. And then, kung matitignan natin itong table na to, for type A, okay, you have antigen A, and then the antibodies found on it is an anti-B. Okay, so that's the determinant na ikaw ay type A. Pag ikaw naman ay type B, you have the antigen B, and then you have the antibody anti-A. For AB, you have antigen A and B, and then you don't have any antibodies. For type O, you don't have an antigen, but you have the antibodies anti-A and anti-B. So the most common one is actually type O, second would be type A, third is type B, and then fourth is type AB. So sabi nga, ang pinakamarami talagang nako-contribute or pwedeng mag-donate ng blood ay mga type O, and ang pinakamahirap talaga na hanapin ay ang mga type AB. Okay? So this is actually how it looks like. So for a type A red blood cells with type A surface, antigens and plasma with anti-B antibodies. So ganyan yung itsura nila, antigen A and then antibody B. So kumbaga parang yan yung mga receptors nila. For antigen B, ayan, yung surface na is ganyan, and then you have the anti-A antibody. And then for antigens A and B, neither anti-A nor anti-B antibodies is present. And then for type O, neither antigen A nor B, but both anti-A and anti-B antibodies are present in the plasma. So guys, yung tinatawag kasi natin agglutination, for example, ikaw ay magpapa-blood typing. Okay? So once na magpatak ka ng isang agglutinating liquid dun sa iyong blood, okay? Pag ang blood na yun ay nag-agglutinate or namuo, okay, ibig sabihin yun ang blood type mo. Okay? So, usually sa isang blood typing, sa isang slide, okay, magpapalagay sa'yo ng 3 dots ng dugo. Yung first one, papatakan siya ng um, antigen A, yung isa antigen B, and then yung isa is to determine if you're positive or negative. So, kung alin doon sa dalawa yung mag-agglutinate or yung mamuo, ibig sabihin yun yung type ng iyong blood. So, kapag walang namuo sa kanila, ibig sabihin type O ka. Okay, even in the positive or negative testing, kapag ka nag-agglutinate, it's positive, and then for negative, hindi siya nag-agglutinate or hindi siya namumuo. Okay? So, this one, the blood typing. Ayan. So, once your bloods are tested, so for type AB, yeah, nag-agglutinate yung first and second. So, your type AB. For type B, ayan, agglutinogen B is present. So, yung B lang ang nag-agglutinate. And then, dito naman type A lang. Kapag type O, walang namumuo sa kanila. Okay? So, fast O's are the universal donors because they have no antigens. Type A can receive A and O blood. Type B can receive B and O blood. Type AB can receive A, B, and AB blood. And then type O can only receive O blood. Okay? So what you see here on your screen right now is actually a representation or a table of which type can actually give to somebody or can receive from somebody. Okay, please study this one as well. Okay, so the RH blood group. So RH factor is simply a protein that is found on the covering of your red blood cells. So if your red blood cells have an RH factor antigen, you are RH positive. If your blood cells don't have this protein, you are RH negative. And 95 to 85% of the population is RH positive. So, antibodies only develop if an RH negative person is exposed to RH positive blood by transfusion or from mother to fetus. Okay? So, if a mother is RH negative and fetus is Rh positive, the mother can be exposed to Rh positive blood if fetal blood leaks through the placenta and mixes with the mother's blood. So first time this occurs, mother's blood produces antibodies against your antigens. Any repeated mixing of the blood can cause a reaction. So next, we have here what we call the hemolytic disease of the newborn. So what is it? It occurs when mother produces an anti-Rh antibodies that crosses the placenta and agglutination and hemolysis of fetal erythrocytes occurs. So this can be a fatal situation for a fetus, and it is also prevented if the mother is treated with RhoGAM, which contains antibodies against the Rh antigens. So this is how the transfusion or the transfer of the Rh can happen. So first one, before and or during the delivery, the Rh positive red blood cells from the fetus enter the blood of an Rh negative woman through a tear in the placenta. So once that happens, the mother is sensitized to the Rh antigen and produces anti-Rh antibodies. And because this is usually happening after delivery, there is no effect on the fetus in the first pregnancy. So during a subsequent pregnancy with an Rh positive fetus, Rh positive red blood cells cross the placenta, enter the maternal circulation, and stimulate the mother to produce antibodies against the Rh antigen. Antibody production is rapid because the mother has been sensitized to the Rh antigen. And then the anti-Rh antibodies from the mother cross the placenta, causing agglutination and hemolysis of the fetal red blood cells, and hemolytic disease of the newborn develops. Okay? So basically with this one, ang nangyayari talaga is that kapag ka na-expose si mother during the first pregnancy, of course, the disease of the newborn develops on the next pregnancy. Okay? Kung baga recurrence ng pagkakaroon niya ng exposure um, doon sa antibodies or anti-Rh antibodies na yun. So, ang nangyayari, nagkakaroon ng agglutination of the fetal Rh positive red blood cells and leads to your hemolytic disease of the newborn. Okay, so nagkakaroon ng pamumuo ng dugo dun sa baby. So, diagnostic blood tests. So, first we have your complete blood count. This provides information such as RBC count, hemoglobin, hematocrit, and WBC count. You also have the hematocrit, which is the percent of total blood volume composed of your RBC. And then the hemoglobin, which determines the amount of hemoglobin and indicate if you have anemia. Okay, so usually these are the diagnostic blood tests that are being asked kapag ka nagpapamedical tayo. So, this one, ayan, paano ba nalalaman yung sa hematocrit number? Of course, you use a centrifuge in the hematocrit tube. Okay, so after drawing the blood, so in the hematocrit tube, you will see there how much is your red blood cells. The form platelets form the buffy coats, and of course the plasma. So next you have the prothrombin time. This is the time it takes for the blood to begin clotting. The normal is around 9 to 12 seconds. And then you have the white blood cell count, so the total number of your white blood cells. So the white blood cell differential count determines the percent of each 5 kinds of your leukocytes. So neutrophils should be under 60 to 70%, lymphocytes 20 to 25%, monocytes 3 to 8%, eosinophils 2 to 4%, and basophils 0.5 to -1. So anything that's more than the normal number, of course, it would suggest a certain disease, okay, that's found in your body. So, other white blood cell disorders, first you have your leukopenia. This is the low white blood cell count. It is caused by radiation, chemotherapy, drugs, tumors, and viral infections. You also have your leukocytosis with high white blood cell count. This is caused by infections and leukemia. So, other disorders of the blood includes erythrocytosis. It is defined as an increase in red blood cell mass, usually absolute, and it is also associated with an increased hematocrit and hemoglobin concentration. You also have leukemia, cancer of the body's blood-forming tissues, including the bone marrow and the lymphatic system. And then you have hemophilia, a rare disorder in which your blood doesn't clot normally because it lacks sufficient blood clotting proteins. So itong mga hemophilia na to, 'di ba? They are actually uso daw or talagang laganap sa bloodline ng mga royalties. Kaya nga ang mga royalties usually talagang inaalagaan sila kasi once na magkaroon sila ng cuts or bruises or um, mga sugat lang, matagal talaga silang gumaling. Okay? And then we have the septicemia. This is a serious bloodstream infection. It is also known as blood poisoning. It occurs when a bacterial infection elsewhere in the body, such as the lungs or skin, enters the bloodstream. Okay. So, thank you very much. That's actually the end of this lecture. So, with the quizzes, of course, be mindful of making sure that you're able to finish your activities on time. Okay, so thank you, and I'll be talking again on another concept that's related with the blood.